This presentation is the genetic alterations, the wrap-up. The topics I'm going to be talking about are the hallmarks of cancer, pathways of tumorigenesis, the role of genomic alterations, environmental factors, and closing thoughts. This first slide is from a January 2022 review, The Hallmarks of Cancer, New Dimensions. This review is the third in a series of Hallmarks of Cancer publications. The Hallmarks of Cancer conceptualization is a heuristic tool for distilling the vast complexity of cancer phenotypes and genotypes into a provisional set of underlying principles. The idea was coined by Douglas Hanahan and Robert Weinberg. In their paper, Hallmarks of Cancer, published in January of 2000, it described 6 biological capabilities and 2 enabling capabilities. The second paper, published in 2011, added 2 more biological capabilities and 2 more enabling capabilities. The table on the right shows the capabilities and a simple analogy. We're going to go through that table now. So one of the capabilities, these are the hallmarks of cancer, the capabilities, is self-sufficiency and growth signals. The simple analogy is that the accelerator pedal is in the on position. The next one is the insensitivity to anti-growth signals. The analogy is the brakes don't work. The next one is evading apoptosis. The analogy is the cells won't die when the body would normally kill the defective cell. Another one is limitless replicative potential. This means that we have infinite generations of descendants. Another one is angiogenesis. The body is being told to give it the tumor cell a blood supply. We have tissue invasion and metastasis. The cells can migrate and spread to other organs and tissues. Over here, we're just giving a color-coded symbol to each one of these hallmarks or capabilities. This slide shows how biological capabilities are acquired during the multi-step development of human tumors. If we look over here on the right, we see a cancer cell, and the cancer cell has all 6 of the hallmarks. And here we can see that there are several different paths to reaching this condition. Note that there is a randomness in the appearance of the capabilities. Each one is different. Notice down here that one molecular event facilitates 2 capabilities right there. Whereas up here, 2 molecular events are needed to acquire 1 capability. So the important message to get here is that there's many different pathways for tumorigenesis. So in 2011, we had 2 new characteristics and 2 new hallmarks that have been added to the picture. Notice that one of them is genomic instability and mutation. The presentations today talked about translocations and mutations, which are part of this enabling characteristic. On the right is the status as of 2022, where we have the addition of 2 emerging hallmarks and 2 new enabling characteristics. notice that one of the new characteristics is the non-mutational epigenetic reprogramming. We've heard about DNA methylation today, and we've heard about microRNA today. These fall into that category right there. So everything that we've talked about today is in this conceptualization of cancer. This next slide shows that intracellular signaling networks regulate the expression of the cancer cell. Subcircuits orchestrate the various capabilities. The various subcircuits are shown in the different shaded regions. For example, here is a mobility circuit. Here is a cell differentiation circuit in the pink. Here is a proliferation circuit, and here's a viability circuit. Note that there is crosstalk between the different circuits. Notice in the center, we see that we have changes in gene expression as a result of alteration in these circuits. These changes then lead to the capabilities, the hallmarks of cancer, which lead to cancer. Proteins are the molecules that are involved in the circuitry right here. So the take-home message to get from this is that complex molecular interactions are involved in the tumorigenesis process. This is just a schematic to show how gene alterations can disrupt normal cell functions that take place in the circuits. So we start with genomic alterations that leads to changes in gene expression. These changes could be an increase or a decrease in protein levels or altered protein structure and function. These then lead to altered cellular function and tumor progression. This slide looks at the role of environmental toxins in cancer progression. So we see that factors like ionization, radiation, and chemical toxins can cause cytogenetic alterations, mutation, and aberrant methylation. These lead to the hallmarks of cancer, the capabilities, and then to cancer. The closing thoughts for this. Tumorigenesis is a complex biomolecular process. This process is enabled by genomic alterations, which can be enabled by environmental factors as well as genetic predisposition. Therefore, it is hard to predict for a single individual the probability of initiating and progressing along the tumorigenesis pathway. Since the year 2000, we have seen significant technology advances in cytogenetic analysis, DNA sequence analysis, DNA methylation profiling, mRNA expression analysis, as well as many other types of molecular analysis. We have seen results from these technological advances in the presentation today. We have learned much from today's presentations, but much more remains to be learned. Our sincerest thanks and appreciation to Drs. Bailey, Kordabash, and Dr. Dubrova for their outstanding work and contributions to this symposium.